Our next presenter is Taz Mueller from the Ecology, Evolution, and Behavior graduate program representing the College of Biological Sciences.
A famous ecologist named John Lawton once wrote, "Community ecology is a mess." And he was right. Understanding communities of organisms is complicated, but it's a foundational question for ecologists. We investigate the who, how, and why of communities, who is there, how they interact, and why a community looks different from another. Now, this doesn't include what a community can do, which we leave for applied fields, but studying the what relies on first knowing the who, how, and why. And when you think about a community, you might picture something like a coral reef or a tropical rainforest. But if you zoom in on that rainforest, inside every leaf, you will find a community of microbes. These are tiny fungi called endophytes, which live inside and benefit plants. Endophytes might be small, but they have huge potential for future applications. We've found that they can help endangered species survive, help crops fight off disease, affect animal and human health through the gut microbiome, and even produce new cancer-fighting compounds. So far, ecologists have figured out who the endophytes are and a lot of how they interact. But the potential applications are still currently limited because the why is still a mess. And this is because to get from a spore to a leaf, endophytes have to go through a gauntlet of 5 main forces. First is distance, how far the spores have to travel to get to a plant host. Second, environment, things like rain or temperature. The third is a host plant's genetics. The fourth is plant traits, which is distinct from genetics because cloned plants can still have different traits. And the last is interactions with other leaf microbes. While this might seem like a linear path, the kicker here is that all 5 of these factors interact with each other, making that linear gauntlet more like a game of chutes and ladders. Distance and environment are usually correlated. A plant's genes combine with its environment to produce its traits. And a plant's traits shape its microbiome, but the microbiome can also feed back to affect plant traits. A good old community ecology mess. And my dissertation research aims to clean it up. I study a California wildflower called Clarkia xanthiana, shown here. This plant's unique environment and genetic structure mean that I can use it to untangle all of these factors at once. For these first 2 forces, I surveyed and sequenced the endophyte communities across Clarkia's entire range. Since its environment varies in patches rather than a gradient, I can separate it out from distance. For the rest, I'm now planting many lines of genetically identical Clarkia in the field. These lines still have different traits, such as leaf color and size, which means I can separate the effects of plant traits from genes. I'm changing their environment through watering treatments, singling it out from a plant's genes and traits. And finally, I'm using fungicide on some plants to suppress endophytes, which will separate out the effect of the microbiome. These treatments cover all the 5 factors and what the communities in each combo looks like. This means that we can finally start cleaning up the community ecology mess of the why to pave the way for the what of endophyte applications. Thank you.
Thank you.